Good morning, Cyprus and everyone online. Thank you for attending my talk. My name is Tisa Loewen, and I am affiliated with Arizona State University in the United States. This presentation is based on my forthcoming dissertation research. Today, I am here to talk about how biological distance analyses can contribute meaningful engagement with the complexities of identity and population dynamics, or, if used incorrectly, perpetuate typological thinking and biodeterminism. Biological distance analysis, or biodistance, is a method of estimating genetic distance between human populations. Today, I'm going to discuss the methodology in brief, provide an example from my research in the Roman Adriatic, and I will discuss both potential and problems. But first, a caveat. I will give an extremely brief discussion of the foundational theory and history behind the method. However, there is so much I cannot cover today, and not everyone thinks about the methodology in this way. And while I disagree, I do not deny there is an enormous breadth of literature that many of us have only scraped the surface of. Some terms quickly. Genotype is the complete set of genetic material of an individual, expressed or not, and phenotype is the observable characteristics of an individual resulting from the interaction of their genotype with the environment. Biological distance analysis is conducted using phenotypic traits as proxies for underlying genetic patterns. These traits are chosen because they provide clues about gene flow specifically. Gene flow is the movement of genetic information through reproduction from one group to another. Common contributors to gene flow are changes to geographic distance, usually from migration or immigration, new interactions that affect the populations that people can mate with. Conflict or allyship can play a role through urbanization, enslavement, or displacement. And culture exchange can affect how people perceive their in and out groups. The traits I use are dental morphology. They are deviations in teeth such as pits, cusps, sizes, and count and shape of those traits as well. They are not arbitrarily chosen. They have specific genetic qualities according to theories developed in quantitative genetics. They need to act as proxies for genetic patterns that suggest gene flow and not the other ways that genes move, that genes which move between people cause their phenotypes to change. The traits need to have three basic properties. They need to pass from parent to offspring in ways that we can observe. Some traits are tied to recessive genes or other complex processes that hide genetic information. In other words, a population can carry a gene but not express it. We don't want to use those. Two, we use traits that are less subject to natural selection or plasticity, something we account for by identifying more persistent traits. So some traits like stature are affected by nutrition and disease, so we avoid those. But over long periods of time, even resistant genes are affected by genetic drift, and natural selection becomes more likely. So, by using a narrow time frame, this can also be accounted for. And we use traits subject to particular population level dynamics that indicate that the variation in the phenotype is proportional to the genes. In basic language, the more genes, the bigger the expression. Together, we refer to these as phenotypes associated with narrow sense heritability, they are neutral, and they are polygenic and additive. The theoretical background was important to explain because without appreciating the mechanisms of the analysis, it is easy to see how the method can be misinterpreted to suggest that certain traits are typological of specific populations, and to mistake that superficial things like appearance are what we are testing for. Also, to show how population dynamics are so important. To further make this point, I will discuss a use case that highlights the importance of cultural context. You can also see the typological sticking points of identity when it comes to naming populations. My research focuses on the late Iron Age and Roman period in the Eastern Adriatic and hinterland of what is predominantly modern Croatia, which is where I collected my data. However, the peoples discussed extend beyond these borders. The initial hypothesis was to see if there were biological relationships between local ancestors and potential descendants who we understand to be Roman. I collected dental non-metric data according to ASU DAS, which is the standard protocol, and I also analyzed them using Anthrop MMD and R package. And the statistic this uses is called the mean measure of divergence. The analysis compares 5 populations, 2 during the Iron Age and 3 during the Roman period. And I present some of the numeric output here, which is what I use. These results do not support any statistically significant differences among everyone examined in the Eastern Adriatic and hinterland during the Iron Age. You can see that in the tan square. Additionally, this analysis did not find differences within groups between ancestors and descendants, which are represented by populations that have 2 time periods. So you can see here we have Pannonia. Geographically, we have Liburnia and Dalmatia, or the home of the Dalmatae. There's an Iron Age sample for the Dalmatia for Dalmatae, an Iron Age sample for Liburnia, and then a Roman sample for each of these, making 5. What is important to mention is you'll notice I pooled coastal populations. So there was a small number of individuals that did not make up their own population that were included in the Liburnian sample. And there were also some Pannonian individuals from the Iron Age that were excluded because that population was too small. And then last, the results show differences between the Eastern Adriatic and hinterland populations during the Roman period. So that's the only statistically significant result here, is that the Liburnians and the Dalmatae Roman descendants, we can call them descendants because we see that the Dalmatae and during the Iron Age and the later Roman period are not statistically significantly different, for example, that the descendants of these two are statistically significantly different. Here is the multi-dimensional scaling figure of the results that you may find more intuitive. The statistically significant difference is between the Roman Liburnians and the Roman Dalmatae. Then I changed the data. I added 6 individuals who were previously excluded and redid the pooling of the populations from one sample to another after considering some of the established history, particularly 1, the Celtic ties in the north, and 2, how the Liburnians' interaction with the Romans when they were coming in the late centuries BCE was very different than the other populations in that they were allied with them, while the other populations had many conflicts and negative interactions and rebellions. But the results for this found no statistically significant differences. So let's look closer. You can see that the results are somewhat shown in this MDS, which is a more intuitive way for looking at the information. We can think about the inputs or variables that went into the analysis, including the context, and how that shaped the results. We can think about the overall variation within the analysis and what effect that is having. For example, one might interpret the coastal Iron Age here sample as overrepresenting coastal individuals' influence, so that by the Roman period, the differences among the descendants stand out. However, you may also notice on the x-axis that they are not on the same scale, so these charts cannot be compared to each other. The analyses are relative. And that is how you want to think about biological distance, as an exploration of the changes in among population variation. Variation can also highlight or draw out both similarities and differences. In this case, I changed the Iron Age samples, and the results were different for the Roman period samples, a result that was not intuitive. So how does this affect us? Well, when used incorrectly and irresponsibly, biodistance can actually enhance and contribute to essentializing populations and making typological conclusions. You can see here on a public forum, people bring up scientific data with varying degrees of understanding and accuracy. We only need to consider American biological anthropology history to appreciate this even more. In fact, it could be said that biodistance is the progenitor of that improper use of biological anthropology, particularly in the late 19th and early 20th century. Researchers have created misunderstandings and distrust in the public's eye and their understanding of science, and at times enhance pseudoscience by providing fuel which we either inaccurately contextualize or misunderstand, which leads to bioessentialism and people using these data for nationalist, prejudiced, and racist purposes. I recognize that European archaeology, and particularly Eastern European archaeology, have their own differences and their own dynamics in history. But ethical science is good science. Romanization is a controversial term, but nevertheless, it is a key component of this discourse in the Adriatic. It has been argued that Romanization creates a false binary when negotiating native and Roman interactions. That dichotomy introduces further complications when talking about population change. As such, biodistance can exacerbate these issues when populations are compared as stable ethnicities. But when done mindfully, biodistance can contribute to better engagement. Inspired by Hingley et al., who state that ideas of ambiguity and temporal distancing can serve to challenge attempts to use such dualities, I recommend that we do not try to isolate the uncertain elements out of analysis, and that we do not present a simple A plus B equals C style of analysis. I find that the proper use of these methods, which introduces the messiness of human complexities, can thwart these very issues instead of contributing to them. Consider the second analysis, we could show, or we could see, that people we understand to be Roman are clearly local descendants as well. The bioarchaeological, biocultural approach can be used to recognize complex and intermixed population dynamics to appreciate the ambiguity. Though relatively straightforward in application, biodistance methods may be perceived as simpler than they really are. So here are some important points to remember. We are not just comparing phenotypes. They are telling us about genes and gene flow. Therefore, we cannot arbitrarily use any phenotype. These are population level analyses, not analyses of individuals. And environment does matter. Also, identifiers are consequential. In the first case, I pooled by geography, and in the second case, I used more detailed historical data. And then, number 5, we are examining shifts in variation. Traits do not belong to or define one population. And that's the end. And here's some select references, and then my acknowledgements, and thank you.